uniQure is probably the longest still existing gene therapy company, and therefore the company has experience in all the aspects of translating something that works very well in a mouse to a real product that benefits patients. We are looking for diseases where there's a clear unmet medical need, and we also look at diseases indeed where a big therapeutic gain can be accomplished by gene therapy. Hemophilia A is a disease that is caused by a deficiency of factor VIII. The reason that you have hemophilia A is because factor VIII is not really activating the coagulation, it's a cofactor for another coagulation factor which is called factor IX. So factor IX doesn't work if you don't have factor VIII. So our approach is different. We now have developed a factor IX that actually doesn't need factor VIII. So we call it super IX. We already have shown that in primates we get sufficient expression of super IX for us to believe that it would have therapeutic benefit in hemophilia A. The next step will be to finalize our toxicology program in primates and all the other work that needs to be done for filing an IND, which will enable us to do a first study in man, and that will be first half of next year, the IND filing. In Fabry, there is a factor alpha-galactosidase which is missing. It's a factor that cells need in their organelles, their lysosomes. They found that there's an enzyme which is very close to alpha-galactosidase, which is called NAGA. The advantage of NAGA is that it's more stable in plasma, that it's better taken up by cells, and that is non-immunogenic because patients with Fabry have normal expression of NAGA. So this we are now developing, and we have done preclinical models already with this, including a Fabry mouse. Our preclinical data have been presented and are quite impressive. uniQure is a result of 10 years of research into the possibility of silencing genes. So in many of our programs, of course, we express a gene, but we also, for some diseases, need to shut off a gene, particularly when the product of that gene, either being an RNA or a protein, is toxic. The trick here is that you silence a gene without causing any adverse effects to cells. These issues have been solved in our Huntington program, and this uniQure technology opens the way to other diseases apart from Huntington, because there are many neurodegenerative diseases that have the same problem. We are very well protected by IP, which is necessary to, for example, manufacture these products. But the other thing is that we have been leading the pack in terms of scaling up manufacturing. We are now in an insect cell system that is readily scalable to large bioreactors. And the plan is to actually further scale up. And this goes hand in hand, of course, with what I started out saying. That is that we are now also able to address larger indications, and you will see us do that in the near future.